All right, so last time we talked about the different processes for material to move in and out of a cell, because we're still discussing the plasma membrane, right? We had the three parts to the cell. We had the cytoplasm, the nucleus, and the plasma membrane. And so we already finished the cytoplasm. Now we're moving on to the plasma membrane. One of the things we learned that it's a limiting barrier, which means it controls what enters the cell and what can exit the cell. So we learned about these processes that there's two types. There's passive processes and active processes. Passive processes is where you're not going to utilize any energy. We don't need ATP. That's the cell's energy source. So we're not going to use any of that. And also, the other characteristic of passive processes is that you're going to see solutes, materials, molecules move from an area of high concentration to an area of low concentration. So that's still going to apply to all the passive processes. So the first passive process that we learned about was diffusion. And we had simple diffusion, and then we just wrapped up facilitated diffusion. So simple diffusion, we see where material would move directly through the plasma membrane. And they had to have two characteristics, really small or just small and nonpolar. So as long as you're small and nonpolar, you can diffuse right through the plasma membrane as long as there's a concentration gradient, which that means is there's more of something on one side of the cell membrane and less of that same something on the other side. Because remember, nature loves equilibrium, it loves balance. Right? So that's what it's trying to do. It's trying to achieve that equilibrium. So we talked about the passive process for diffusion. And then we learned what facilitated diffusion is. It's still a passive process, no energy is utilized. We're still going to move down a concentration gradient. But now we're going to need something to help facilitate that movement of the solute. And so what did we use? Transport proteins. And we just talked about, come on, why aren't you working? We talked about channel-mediated diffusion as one of them. We learned about the leak channels and the gated channels. One's the 7-Eleven, always open. The gated channel, that is going to be closed unless a stimulus causes it to open up. And so we talked about, we'll see a lot of this when we get into our later chapters, especially your third lecture test, which covers chapter 10 on muscles and chapter 12 on nerves. So we're going to see a lot of that later on. But I just wanted to drop that information on you just to kind of help you out. And there's an example of our channels. And so you'll see there's lots of sodium outside the cell, not a lot inside the cell. And so sodium wants to go to the area where there's less of it. So it moves from the area of high concentration to the area of low concentration. And vice versa here with potassium. Lots of potassium inside the cell in the cytosol. Not too much of it outside the cell in the interstitial fluid. So potassium leaves the inside of the cell and goes outside the cell. All right, so that's basically diffusion. You're going to move down the concentration gradient. It seems like I'm saying that a lot. It's because it's important. And then we talked about the carrier-mediated diffusion. Here we have this special transport protein that's going to do the same thing. It's going to move an item from an area of high concentration to an area of low concentration. The example here is glucose. All right, a lot of glucose outside the cell, not too much of it inside the cell. And so this special transport protein is going to help facilitate or get it to move from that high concentration area outside to that low concentration area inside. So now we're going to move on to another passive process. And there's a couple things I want you to know. Some of us are familiar with osmosis. My roommate in college felt that they could learn better through osmosis. And his idea was sleeping with his textbook under his pillow at night. Did not work, all right, for many reasons, all right? But according to our definition of what osmosis is now instead of dealing with solutes with osmosis, we're going to deal with water only, okay? So it only involves the movement of water. So if you're ever taking a test in this class and something in that question that you're reading mentions osmosis and what it's moving, your answer choice has to involve water. It has to. Okay, so technically the osmosis is the movement of water through a semi-permeable membrane from an area of high concentration to low concentration. So in our class here, for our purposes, what do you think that semi-permeable membrane is going to be? The cell, but what part of the cell? The plasma membrane. Yeah, that's our semi-permeable membrane. Okay, so we're going to apply osmosis now to the plasma membrane. And so that's what we're talking about. You could say semi-permeable. That's one of the common definitions. You can also say selectively permeable, right? But similar, okay? We're going to be going through a membrane that's going to let some things pass, but not others. And in this case, we're going to deal with water. So the same type of rules apply. It's a passive process, means no energy is going to be utilized during this process. And second, it's not the solute that's moving, it's water that's moving, but it's going to move down the concentration gradient, which we've already learned. So if you have a lot of water outside the cell and not a lot of water inside the cell, which direction does water want to go? Right, outside to in. That's what we want to do. Okay, so that's all what we're going to see. So we're going to see, okay, our plasma membranes are going to allow, right, water to move across the cell. And even in some cases, right, you're going to see that special transport proteins, we call them aquaporins. The cell will manufacture these things and put them into the plasma membrane so it's easier for water to move across the plasma membrane. And you're going to learn about some of those cells in your kidneys, depending on if you're in the hydrated state or the dehydrated state. And we'll learn about that. All right, so keep in mind, and this is a very important concept, your plasma membrane is going to prevent the passage of specific solutes. Example, sodium. It prevents the passage of sodium because it's charged. Right? And so, how does sodium get across the plasma membrane? Through facilitated diffusion. We have a special transport protein that allows for that. So we'll see that. Same thing with glucose. Glucose just can't go through the plasma membrane. Oxygen can, because it's small and nonpolar. Carbon dioxide can, because it's small and nonpolar. You got to meet those criteria to go right through the plasma membrane. All right, so that's what we're going to be talking about here for osmosis. So the two ways that we get across is to go directly through that phospholipid bilayer. So you can say it kind of slides in between those molecules, those phospholipids. Or, like I said, we're going to use those aquaporins there, those specific protein water channels that were built by the cell, installed into the plasma membrane that lets water move across. So when we're talking about solutes, and solutes is just a general term that we're going to use for whatever it is that we're discussing that's going to move across the plasma membrane. All right, so we have permeable and non-permeable. Right, so the permeable ones, obviously the term permeable, that means that that solute is going to pass right through that phospholipid bilayer. And so they usually have these characteristics. We know this already. We've talked about this. Small and nonpolar. Our non-permeable solutes, they can't just diffuse right through the plasma membrane. Okay? And so what characteristics do they have? Charged. Okay, what's charged? What's a charged molecule? Well, the ions. Remember the cations and the anions? Okay, so ions are charged, all right, molecules, so they can't pass through, so it's non-permeable, right? If you're polar, or if you're just really, really big. Glucose is big. Proteins can be really, really big. Right? So they need help. So you want to know the two types of solutes that we're dealing with here. Permeable, all things are all good. You're small, nonpolar. You just slip and slide in between those phospholipids, and you've gained entry into the cell, or you've managed to exit the cell. Permeable, different story. You see those characteristics. They're charged like an ion. If you're polar, or if you're a large solute, like a big old protein, can't just slip in between those phospholipids. You have to get into a cell or out of a cell a different way. Kind of good with that? Okay. We're still on water though, okay, because it's osmosis, but I just want to kind of talk to you about those different types of solutes there. Okay. So let's talk about, remember, it's a passive process, so we're going to be dealing with no energy. I know I keep repeating myself, but I want to drill that into you. But we're going to deal with our concentration gradient here, right? So we have to decide the concentration gradient for water. And it's going to be between two areas. Outside of the cell, which is what we call the interstitial fluid, can also be referred to as the extracellular fluid, ECF. And then the other area that we're going to be discussing is what we're going to be discussing is the inside of the cell, right? And that's the cytoplasm, but specifically, we're going to be talking about the cytosol, which is the fluid inside the cell. And the majority of the cytosol is made up of what? Is it up there? When in doubt, when we're talking about fluids, always guess water. Always guess water, okay? I mean, you've heard people say, oh, your body's 90% water, okay? Part of that's true. Now, I won't say part of that's true. I mean, that's pretty extreme. But point being is the majority of a lot of these fluids in your body, and you'll learn it when you talk about the blood in chapter 18 and 211, but it's going to be water. All right. So we're going to talk about the movement of material between those two spaces there. Keep in mind, right, depending on the type of solutes that we're dealing with, some of those solutes are going to be prevented from crossing the plasma membrane if they're charged, if they're large, if they're polar. Right? So they can't go. Right? Water's a different story here. So we're going to discuss what causes that water concentration gradient to exist here, and then what happens to the solutes. And we'll discuss what I mean by that, because when it comes to osmosis, solute concentration will play a role in what happens with the movement of water. And I'll touch on that in a little bit, okay? I'll touch on that in a little bit. For right now, I want you to know water with osmosis, it's going to go from an area of high water concentration to an area of low water concentration, all right? Okay, so let's talk about that movement of water. So like I said, it's based on that concentration gradient between the cytosol and the interstitial fluid. And so wherever there's more, it's going to move to the area wherever there's less there. So keep that in mind, and we don't change the rules. Water is still going to move down that concentration gradient until we get equilibrium. Okay, equilibrium. And so the example here that we talk about, water is going to move from a solution of 1% solutes, right, into a solution containing 3% solutes. So if you notice this example here, water moves down its concentration gradient, and then you can see here, water moves from a solution of 1% solutes to an area of 3% solutes. So you see it. It's going to go from an area of low solute concentration to an area of higher solute concentration. That's interesting. Okay, that's very interesting. So keep that in mind. I'm going to touch on that more here in a few moments. But the area that has less solutes has higher water concentration. And the area that has a higher solute concentration is going to have a lower water concentration. For right now, just memorize that. So an area that has more solutes will have less water. And an area that has less solutes in it, right, will have a higher water concentration. And I'll show you why. All right, I'll show you why. We'll talk about some of those concepts. But kind of just right now, just memorize it. We'll touch back on that in a second. All right, so when we're looking here at our cell, there's a lot on this slide here. I'm going to zoom in on it a little bit. And this is what we're going to see with the movement of water in and out of a cell. So first of all, let's establish what we're looking at here. Here's the plasma membrane. There's our selectively or semi-permeable membrane. Here's the outside of the cell, or excuse me, this is the outside of the cell, the interstitial fluid. This is the inside of the cell. That's going to be the cytosol, also known as the intracellular fluid. And so you can see we have our plasma membrane, the phospholipid bilayer, and then we have some of these transport proteins that we've inserted into the plasma membrane here. So the water molecule is going to be designated by these red balls with the two, those look like light blue bumps on them. And that's representing H2O, okay? Oxygen is the red molecule, and then the hydrogen are those two little white or light blue bumps on it, okay? So notice On which side of the plasma membrane do you see more water molecules? Okay, so on the right side, right? Which would be the cytosol, right? Yeah, the intracellular fluid. Good, good. Okay, so on the outside, you see a couple out there. So which side has the higher water concentration? The right or the left? The right. Okay, you notice that the left side has very little. So knowing what we know and what I've discussed about osmosis so far, where does water want to go? It wants to go to the area with the lower water concentration, which would be the left side of that plasma membrane. That would be the interstitial fluid. So it's leaving the cell. And you can look at the arrows. When it's going through the aquaporins, it's leaving the cell. And then you can even see when it's going through the middle here of the phospholipid bilayer, it's leaving the cell. Would we all agree on that? So it's following our rule, right? It's going down its concentration gradient. So you'll notice then in this area with the water, where there's a lot of water on the right side of our plasma membrane, you got some other things in there. We got some proteins, we got some glucose, right? We have some other molecules like potassium, for example, right? But if you look and compare all of those other molecules that are non-water, let's say, and then compare it to all of these molecules on the outside of the cell, which side would you say has more of those non-water molecules, what we call solutes? Left or the right? The left, okay? So you can see where water is going to the area that has the higher solute concentration. Okay? This is what we just talked about in this previous example here. All right, so water is going to go from the higher water concentration to the area of lower water concentration, but we can also say that water wants to move to the area where there's a higher solute concentration. That's very important later on. So memorize that and store that away for right now, okay? So water moves from an area of high water concentration to low water concentration, or you could say water is going to move from an area of low solute concentration to an area of high solute concentration. That's going to influence, right, the movement of water. So that brings me to our next concept, which is called osmotic pressure. So the definition up there says pressure exerted by movement of water across a semi-permeable membrane. That's our plasma membrane. So as these water molecules are moving, okay, they're going to create this osmotic pressure, right? And so what created this osmotic pressure? This part right here. Let me underline it for you. Difference in the solution concentration. So what's that difference in the solution concentration? I'm going to come back to this slide, I promise you. The fact that there's lots of solutes in our example outside the cell and fewer inside the cell. The fact that there's an unequal distribution of solutes across the plasma membrane. That's what's going to help to create this osmotic pressure. So if there's a large difference in solutes, if there's 100 molecules outside the cell and maybe 2 inside the cell, that's a large difference, right? 100 minus 2 is 98. But if there's like 50 outside the cell and 49 inside the cell, it's only a difference of 1. So your concentration gradient, right, whether it's steep or not, that the steepness of that concentration gradient is going to determine how much water, that pressure of water, all right, is going to be generated. So if you have a not so steep pressure concentration gradient, then not a lot of water is going to move. But if it's a really steep gradient, kind of like what we see in this picture here, we're going to see a lot of water moving from that area of high water concentration to that area of low water concentration here. And so this moves into this concept called hydrostatic pressure. Now, I promise you, you will revisit this concept a lot when you get into chapter 20, when you talk about the circulatory system. It's just not in this class, okay? But I want to introduce you to the hydrostatic pressure, right? So the definition that's listed up there is the pressure exerted by a fluid on the inside wall of its container. Think of water in the pipes in your house. when the water's inside those pipes, it's pressing on the internal surface of the pipe. Same thing goes on in your blood vessels and in your lymphatic vessels when we're talking about blood. When it's moving through your veins, when it's moving through your arteries, blood is pressing on the walls of those vessels, and it's pressing outwards. So when we're dealing here with osmotic pressure here, we're still going to be, water plays a role in that. So your plasma, which is the fluid part of your blood, 92% of it is made up of water. So the majority of that fluid is made up of water. And so when it's flowing through your blood vessels, you can say it's trying to get out. All right. But of course, your bigger blood vessels, like your arteries and your bigger veins, keep that water in. It's not going anywhere. But when we get to these really small blood vessels, they're called capillaries. Capillaries can have one cell layer thickness for their wall. It's not like simple squamous epithelium. In fact, that's what it is. The capillary wall is simple squamous epithelium, but it's only one cell layer thick. So water can leak out of there, right, due to this hydrostatic pressure. And that hydrostatic pressure changes depending on if there's more solutes inside your blood or if there's more solutes outside your blood. For example, say there's lots of solutes outside the blood, 99 molecules outside the capillary wall and only one solute inside the capillary wall. Where does water want to go? Towards, so to where it's higher, so it wants to leave the capillary, right? Because there's 99 molecules outside, one's inside, so it wants to leave. So if we were to change that and say that there's 99 molecules of solutes inside your capillary and one molecule outside, then does water want to leave the capillary or does it want to stay where all those solutes are? Yeah, it wants to stay. It won't leave. So again, your solute concentration, wherever there's more solutes, water, I say this to my other classes, water is a follower, right? What's one of the characteristics of a person with diabetes? What do they complain that they do a lot of? Pee. Why? What's special about their pee? What do they usually say? What do we say about their pee? When you do a dipstick test and you're testing their pee for something, well, there's ketones. That's a great one. Yep, that's not what I was thinking, but ketones is one. Well, it's a solute, so you can consider that. But what's the big one? What does everyone say? Sugar. Sugar in the urine. Glucose. You should never be peeing out glucose. Not a single person in this room should ever be peeing out glucose unless there's something going on. Okay? So what happens is a person with diabetes pees glucose out. Right? But that's not what's going to give a large volume to your urine. What's going to give you a large volume to your urine is the fact that as you are excreting glucose in your kidneys, you're going to also be excreting water because water follows glucose. So in this example, glucose is our solute. What's one of the things that they say to us about people that have high blood pressure? What do you want to avoid in your diet? Sodium, salt. Exactly. So they say don't eat salt because sodium is one of the components. We know what sodium is. Salt is sodium chloride. So sodium is also a solute, and water loves to follow sodium. So if you pee sodium out, then you're going to pee, water's going to follow it. So they say to us, don't take in sodium in your diet because sodium is eventually going to wind up in your blood. And if you increase the concentration of sodium in your blood, remember what I said, water is going to follow sodium. So then water is going to move into your blood circulatory system, and that's going to increase your blood volume because the water starts to increase the overall volume. If you increase blood volume, you increase blood pressure. It's an important concept to know. I won't test you on it, but when we get into 211, if you decide to take me, if you're still happy with me as a teacher, I cover that quite extensively. So the point is, where solutes go, water follows. So if there's a lot of solutes in compartment A, then water wants to go to compartment A. Right? If there's a lot of solutes in compartment B, water wants to move to compartment B. So you can kind of think of it like that. And keep in mind, where your solute concentration is high, your water concentration is low. It's almost like an inverse relationship and vice versa. So I'll show you a picture that will kind of explain that. right? But keep that in mind about what osmotic pressure is. It's just that pressure that water exerts as it's moving across the plasma membrane. And what influences that pressure? Solute concentration, okay? And especially if it's a big difference, if it's a steep concentration gradient. So that's what this picture is showing us. So we have our little horseshoe structure here. And so if you look at it here, right, here's our horseshoe water container with our semi-permeable membrane that only water can cross. Now, if you look here, side A, these black balls, those are the solutes. So let's count them up. 1, 2, 3, 4, 5, 6, 7, 8 on side A. And then look at side B. Let's count the solute number there. 1, 2, 3, 4. So before we even look further, there's more solutes in side A, less solutes on side B. Where does water want to go? It wants to go from side B to side A because there's more solutes. It wants to go where there's more black balls there. Those are the solutes. So we have 8 here, 4 here, more solutes here, less solutes here. But as a consequence, understand, if you have a higher solute concentration, you have a lower water concentration because they take up space. You can't have two things. It's physically impossible. You cannot have two things that have mass occupy the same space. So if you have a lot of solutes, that means you don't have enough room for water, so you'll have lower water concentration. So here, higher solute concentration, lower water concentration. So what's osmosis? Movement of water down its concentration gradient from an area of high water concentration to an area of low water concentration. And if you want to count it up, you can. You can count up the red balls. There's going to be less red balls here on side A than there are on side B. So when water decides to move through that semi-permeable membrane, whoops, it's going to look like this. So now water has flowed through that semi-permeable membrane to the area of, at one point, had a less water concentration. But you can see it's changed the configuration here. It looks like there's more water here. Right. And it's only going to move until it reaches its equilibrium. So I just want you to get those two concepts down. Water moves from an area of high water concentration to an area of low water concentration. And so if we look at our picture here, in previous classes, I've counted up the red balls, but you can see more water molecules on this side, less on that side. So water goes from high water concentration to low water concentration. But at the same time, there's more solutes on this side than there is on this side. So you can also say water moves from an area of low solutes to an area of high solutes. It almost kind of pulls it in. Because remember what I say, water is a follower. It's going to follow certain solutes more than others. So that leads us here to this concept of osmosis and what we call tonicity. So going back to our cells, right? Cells are constantly gaining and losing water depending on what's happening with the solute concentration there. Right? It happens all the time, whether you're dehydrated or whether you're hydrated. So when water moves out of the cell, your cell volume shrinks down. Less stuff inside, so it gets smaller. When water moves into the cell, you increase, right, that cell volume there. It's like a water balloon. Okay, you keep filling up that water balloon with water, eventually it can pop, but you're increasing that water volume inside the balloon. And that's what happens to your cells quite often. So when water moves in and out, we change the cell volume. At the same time, when water is moving out, we're going to change the osmotic pressure. Because remember, osmotic pressure is that pressure that's generated, right, with the movement of water. So that leads us to this term here called tonicity. Right, so the ability, and you want to memorize this, the ability of a solution to change the volume or pressure, that pressure we refer to as tone, hence the term tonicity, of a cell by osmosis. So basically what that's saying is we can influence the cell's volume with the movement of water. Like I just said before, if water enters into the cell gets bigger. If water leaves the cell, it gets smaller. And as water is moving in and out of the cell, that is going to influence or change the pressure, the osmotic pressure, because that's all. Anytime you get water movement, you have osmotic pressure that's being generated. So more water movement, more osmotic pressure. Less water movement, less osmotic pressure. So we're going to talk about these three possible solutions that you can be dealing with. Right? So there's three types of solutions that we're going to be discussing that can influence the movement of water. So the first one up there is isotonic. Then the next one is hypotonic, and then there's hypertonic. Isotonic, nothing's really changing. Hypotonic, you have decreased tonicity or pressure. And then hypertonic, you'll have an increase. And usually that's in relation to when we talk about the tonicity of a solution, it's based on the solute concentration. Because remember, does not the solute concentration influence the movement of water? Lots of solutes, lots of water movement. Not a lot of solutes, not much water movement. Okay? So there's that relationship that occurs there. So those are the three, and we're going to go through these now here, and I'm going to show you examples. So the first one is called isotonic. Right, so this is just, I apologize, but it's just straight up memorization on this one. So isotonic, just like if you've heard of isometric muscle contractions, that's when you're flexing your muscles like bodybuilders do when they're on stage, but there's no movement going on. Right? So with isotonic, there's not going to be any movement going on because both inside the cell and outside the cell, in this case, the solution, have pretty much the same number of solutes. So if you have the same number of solutes inside the cell and the same number of solutes outside the cell, what's water going to do? It's going to stay in place. It's not going to go anywhere, right? Because it has equal amounts. And in that case, normally then you'll have equal amounts of water then on either side. So when you have equilibrium, everything is, I won't say happy, but you're not going to see much occurring there. So here's a great example. Normal saline, right? Normal saline is used in IVs when folks have lost blood for whatever reason. Okay, they'll hang it if you need to increase that blood volume. Remember what I said, if someone's blood pressure starts to drop, for example, maybe they lost their hand and they were bleeding out, right? So, of course, a lot of bad things can happen when your blood pressure starts to drop. We want to maintain, remember homeostasis, we want to maintain that normal blood pressure range. 140 over 90 is the high end, 90 over 60 is the low end. We want to keep you in that range there, one, to save your life, right, as you're bleeding out. So what do? What did I say? If you increase your blood volume, you increase your blood pressure. That's essentially what they're doing when they're giving you normal saline. They're increasing your blood volume to help maintain or increase your blood pressure there. All right, so in this case, you want to give people, all right, an isotonic solution because we don't want water leaving the blood system, the circulatory system, right? We want to keep it there. So that means if we're talking about blood, we're going to talk about the red blood cells. Those are the erythrocytes. This type of solution, when you take a red blood cell and dump it into an isotonic solution, nothing's going to happen. Water won't leave the cell and water won't enter into the cell, which is good. Because I'm going to show you now what's going to happen when we put a red blood cell into a solution that's either hypotonic or hypertonic. So you're going to see what happens here. So let's start off with the hypotonic. Okay, so here's the definition of a hypotonic. The solution has a lower concentration of solutes. Less solutes outside the cell, more solutes inside the cell. So what does that mean? That means that there's more water outside the cell because we have a low solute concentration. And then we have a low concentration of water inside the cell. That's what we're kind of looking at. I'm going to give you all that information there. So, of course, we know, what does water want to do when we're dealing with osmosis? It wants to move down its concentration gradient. And in this case, you're going to have more water outside the cell, less water inside the cell. So where does water want to go? It says it up there. Okay? It wants to move from outside the cell to inside the cell. So guess what we're doing to that red blood cell? If we put it in a hypotonic solution, yeah, we're filling it up like a water balloon. Okay? And guess what happens as water enters into the cell? The inside of the cell becomes larger, so we increase the volume of the cell. All right, and also at the same time, we're increasing the pressure. Feel a water balloon sometime. If you didn't fill it up too much, it doesn't feel that firm. The balloon won't feel that firm. But when you start to fill up that water balloon to the point where it almost is going to pop, feel the water balloon. All right, it feels a little bit more tense. All right, and that's because we've increased that water pressure. So here's what's going to happen. Water keeps entering the cell. It keeps getting bigger. You keep inflating your water balloon with water, and it eventually will what? Pop. And we call that lysis. Right? Lysis is when you get the rupturing of a cell because the inside of that cell's pressure has gotten so great, right? And the difference, right, in that water pressure and that osmotic pressure has become so great it causes the cell to burst. When you do this to red blood cells, we call that hemolysis. Heme has to do with blood, red blood cells specifically. And so that's what's going to happen. So if you take in something that's hypotonic and it gets into your circulatory system, you will start to see the rupturing of red blood cells. Right? And so we want to avoid that type of scenario there. All right, the last type of solution is going to be the hypertonic. Right? In a hypertonic solution, that means that the solution, which is the area outside of our red blood cell, has a higher concentration of solutes than inside the cell. So what did we say? Where does water want to go? It wants to go inside. It wants to go outside the cell because there's higher solute concentration, right? And that's what we're going to see. So when you give somebody or you put a red blood cell into a solution that has 3% sodium chloride, right, outside the cell is going to have a lot of solutes. So the water that's inside that red blood cell is going to leave the cell. And so when it leaves the cell volume decreases, the osmotic pressure inside that cell decreases, and that cell essentially shrivels up, or it starts to shrink. And so we call that crenation. We call that crenation. And again, that's because water goes from an area of low solutes to an area of high solutes. Higher solutes outside the cell in the solution, so it's going to leave the cell and go outside, and it causes the cell to shrink. So that's what it looks like in these pictures here. Here's our isotonic solution. We drop our red blood cell into it. There's equal amounts of water outside the cell, equal amounts of water inside the cell, because there's equal amounts of solutes outside and inside the cell. So no movement whatsoever. Now, when we go to the hypotonic solution there, right? So less solutes outside the cell, more solutes inside the cell. So water is going to move, right, from the low solute concentration area to the area where there's a higher concentration of solutes. So it's going to move into the cell. It's going to follow. Wherever the solutes go, that's where water wants to go. And the cell starts to build up, and then it pops. And then finally, we have our hypertonic solution. Lots of solutes look outside the cell and not too much inside the cell. So water is going to go to where there's more solids. And so it leaves the cell, and the cell starts to shrink. So what occurs to the tonicity of a cell when placed in hypertonic solution? There you go. There's your hypertonic solution there. Okay, so that's what you're going to see. You're going to see water leaving the cell, which causes a decrease in the cell volume, which causes a decrease in the pressure inside the cell, causing it to shrink. This may have been the longest I've talked about this subject, all right, to a class, but I really wanted to nail those points down for y'all. Okay, that's perfect. This is where I want to end things here.